Hello, I'm Victoria Unesbem, Manager of Nutrition Science and Education and Summit Planning Committee member here at the ANA. I'm thrilled to be introducing Dr. Artemis Simopoulos, who will be joining us today to discuss genetic variation in polyunsaturated fatty acid metabolism, inflammation, resolution, and COVID-19 risk. Dr. Simopoulos is the founder and president of the Center for Genetics, Nutrition, and Health, a nonprofit educational organization in Washington, D.C. She was a founding member of the International Society for the Study of Fatty Acids and Lipids, ISSFAL, and she is the founder and past president of the International Society of Nutrigenetics, Nutrigenomics, ISNN. She is a physician and endocrinologist whose research has focused on nutrition in genetic and endocrine disorders, the evolutionary aspects of diet with a particular interest in fatty acids, especially omega-6 and omega-3 ratios. Dr. Simopoulos has held prestigious public roles. She was chair of the Nutrition Coordinating Committee at the NIH and consultant to the Office of Consumer Affairs at the White House. Prior to that, she was the executive secretary of the Division of Medical Sciences at the National Academy of Sciences. She is the author of the Omega Diet and has edited over 50 books and journal supplements, in addition to publishing over 360 scientific papers. She was the editor of the Karger series, World Review of Nutrition and Dietetics, for over 2 decades. Dr. Simopoulos was the American College of Nutrition's 2018 recipient of the Humanitarian Award for her selfless work in the field of nutrition. And before she begins, it is my distinct pleasure to recognize Dr. Simopoulos with the 2021 Selig Award. This award, given in memory of Drs. Alexander and Mildred Selig, recognizes a highly esteemed scientist who has made outstanding contributions to the field of nutrient research. Congratulations, Dr. Simopoulos, on this well-deserved award.
Hello, everyone. You can see my faculty disclosure and the presentation learning objectives. And now I would like to start my presentation. I will begin with certain fascinating historical aspects. In 1976-77, the McGovern Committee on Nutrition and Human Needs or Human Health was very active in Congress, and they developed the report, which is called the Dietary Goals for Americans. The report was sent to the various federal agencies for comment, as it is customary. At that time, I was the chair of the Nutrition Coordinating Committee at the NIH, and it fell upon me to pull together the comments from the various institutes and draft a letter of response to Senator McGovern. We were very concerned that they thought they could have the same goals for everybody. And we pointed out that it's not the appropriate terminology and that dietary guidelines are more appropriate. So we coined the term dietary guidelines. The letter ended with the following paragraph: should increased emphasis on prevention of disease and the promotion of good health ever become more than a pious hope, analysis of each genotype will be an essential prelude to guidance toward and away from particular conditions known to enhance or to threaten the individual's adaptive state. The genetic view, which predicts that not everyone is equally susceptible to all threats, suggests that non-susceptible persons should be spared the fearful anticipation of events that never materialize and the requirement to abide by rules that are for them irrelevant. So, although they accepted our term of dietary guidelines instead of goals, NIH really never developed a nutrition research program that included genetics. So, what a pleasant surprise, on August 25th, 2020, the New England Journal of Medicine carried an article under the term Precision Nutrition, the answer to what to eat to stay healthy, by Dr. Rogers and Dr. Collins, the director of the metabolic diseases, diabetes metabolic diseases, Dr. Rogers and Francis Collins, the director of NIH. And they define, like precision medicine, the field of precision nutrition aims to understand the health effects of the complex interplay among genetics, please note it's in capitals and it's the first area, microbiome, antibiotic and probiotic use, metabolism, food environment and physical activity, as well as economic, social, and other behavioral characteristics. So there is about 44 years between the time that we responded to Senator McGovern, emphasizing the importance of genetics in nutrition, and when NIH moved in to support nutrition research and develop a strategic plan that has 4 major pillars: what to eat and the effects of food and nutrition, what and when to eat, how does nutrition promote health across the lifespan, and how to improve food as medicine. How to improve food as medicine fascinates me because this is roughly what Hippocrates said in 480 BC. Let food be thy medicine and medicine be thy food. So it took a long time, but there was progress. Hippocrates defined the concept of positive health as follows. The emphasis, of course, was on prevention, but the word prevention has a negative connotation in Greek, so he referred to it as positive health. Positive health requires a knowledge of man's primary constitution, what today we would call genetics, and of the powers of various foods, both those natural to them and those resulting from human skill. Today's processed food. But eating alone is not enough for health. There must also be exercise, of which the effects must likewise be known. The combination of these 2 things makes regimen, when proper attention is given to the season of the year, the changes of the winds, the age of the individual, and the situation of his home. If there is any deficiency in food or exercise, the body will fall sick. This very important concept is being represented by this triangle originally established by Barton Childs, with whom I worked very closely. So the relationship between genes, environment, and development are dynamic, and our phenotype, what actually we become, is the result of the interaction between our genes and various environmental factors, nutrition being an environmental factor of major importance. So I would like to begin now, having given you the history of the events that led to today's precision nutrition program and research activities at the NIH, by looking at the evolutionary aspects of diet, genetic variation, omega-6 and omega-3 fatty acid biosynthesis. Human beings evolved on a diet that was very different from today's diet. Major changes began to take place with the development of agriculture 10,000 years ago, but more changes occurred following the industrial development, and particularly the time after the Second World War. Major changes in the diet of animals, being grain-fed rather than grass-fed, produced meat that is high in omega-6, both in terms of linoleic acid and arachidonic acid, and a practical deficiency of omega-3 fatty acids. But more changes came about when oils were made from seeds, and oils from corn, like corn oil, sunflower oil, soybean oil, cottonseed oil, became very popular, both in terms of food processing as well as cooking oils, with restriction of butter. Margarines followed the oil production, which of course was a major mistake because the trans fatty acids in margarine are extremely dangerous to health, and we no longer have them in high amounts in our food supply. So what you see in this slide is the fact that the omega-3 fatty acids shown in blue really began to fall about 70 to 90 years ago. And the omega-6 fatty acids already began to increase just before the Second World War, with enormous increase after the Second World War. And then the same increases occurred in saturated fat and total fat, where the antioxidant vitamins like vitamin C and vitamin E decreased. So there were major changes in the food supply, not because of scientific reasons, but because the technology was there. And there was a number of human studies that show that it's possible to increase the biosynthesis of EPA, DPA, and DHA from alpha-linolenic acid, the omega-3s, but you need to reduce the linoleic acid to a level below 2.5% of energy, which will be very difficult to do because the LA in the food supply in Western diets, including the American diet, accounts for 6 to 8% of energy. And such levels reduce the production of DHA. So not only we don't eat as much fish as we eat meat, not only we use oils high in omega-6, but we also reduce the production of DHA from alpha-linolenic acid because of the high amounts of LA. And of course, the biggest problem that we have is, adding insult to injury, the American Heart Association recommends that LA increase to 10% of energy, which is totally unscientific, inappropriate, and dangerous to health. Studies show that LA increases have lowered the omega-3 index by 41%, from a 6.51 to 3.84. Anything in the omega-3 index of less than 4 is considered deficiency of omega-3 fatty acids. And I will show you at the end, most of the world has an omega-3 index of less than 4%. Modern Western diets, including the modern diet of Japan, as a result of moving away from traditional diets, have led to omega-3 deficiency syndrome. In 1997, Okuyama summarized the evidence, which indicates that increased dietary LA and relative omega-3 deficiency are major risk factors for Western-type cancers, cardiovascular and cerebrovascular diseases, and for allergic hypersensitivity. Studies also suggest that the omega-3 deficiency created by excessive intake of linoleic acid and high LA to ALA ratio, a high omega-6 to omega-3 ratio, by Western diets affects human behavior in industrialized societies. So the omega-6 and omega-3 long-chain PUFA and their metabolites have structural and signaling roles throughout the human body. So any change that varies from our evolution aspects is not consistent with health. Maintaining a proper balance of the omega-6 and omega-3 long-chain PUFAs and their metabolites is critical to homeostasis in virtually every physiologic system. Therefore, environmental and genetic mechanisms that influence their levels and balance will impact human health and disease. I now like to focus on the biosynthesis of omega-3 and omega-6 fatty acids. The omega-3 PUFA are shown in green, and the parent fatty acid is alpha-linolenic acid, which is 18 carbon atoms, and via the enzyme of delta-6 desaturase, as well as elongase and the delta-5 desaturase, it actually forms eicosapentaenoic acid. Eicosapentaenoic acid is the parent fatty acid for the biosynthesis of eicosanoids. The term eicosanoids because the metabolites consist of 20 carbon atoms. Eikosi is 20 in Greek. So through enzymes such as COX-2, 5-LOX and 5-LOC, we have the production of a series of thromboxanes and leukotrienes that are less thrombotic and less inflammatory than those that are produced by the omega-6 fatty acids. Again, the omega-6 fatty acids, the parent fatty acid is linoleic acid. It also uses the same enzymes for the elongation and desaturation as those of the omega-3 series that goes to EPA. And we have the formation of arachidonic acid, which is 24 omega-6. Again, just like EPA, the arachidonic acid is the parent fatty acid for the production of eicosanoids of the 2 and 4 series. The eicosanoids from arachidonic acid are prothrombotic and pro-inflammatory, except for lipoxan A4, which is the only product of arachidonic acid that is less inflammatory. So both pathways, the omega-3 and omega-6, use the same enzymes, but through evolution, the omega-3 and omega-6 were balanced, and the omega-3 PUFA metabolism was the preferential one. But what happened because of changes in the food supply, we have enormous amounts of omega-6, so we produce a lot of arachidonic acid, and this becomes the preferred pathway as a result of the Western diets that are high in the oils of omega-6 fatty acids. Now, the delta-6 and delta-5 desaturases are used by both pathways, as are the elongases. So genetic variants exist at all the enzymes throughout the metabolism. And it has been noted that African Americans and Hispanics, they have higher amounts of arachidonic acid, much more than you would expect from the diet. And these high amounts of arachidonic acid are found in the red cell membranes phospholipids and in all the tissues. And you can see that studies in African Americans, they show the arachidonic acid is much higher in African Americans than in Europeans, and the same with DHA, but not as much. The DHA is in European Americans, it's much lower than what it is in African Americans. So the question is exactly what is going on besides the diet? What else could increase the high amounts of omega-6 fatty acids in African Americans and in Hispanics? So there were a number of studies, and people looked for genetic variants, and Amer did very extensive studies and found out that there are 2 haplotypes that are involved in the formation of arachidonic acid from linoleic, as well as the EPA and DHA from omega-3 fatty acids. So these two haplotypes are called derived and ancestral. And the ancestral haplotype is the one that was very prominent about 80,000 years ago. But what happened after that, there was a lot of movement. The populations in Africa moved away from the seashores into the savannas and in landlocked areas. And so they really did not have that much fish to eat in order to have enough DHA for the brain. And they adapted by having a much more rapid metabolism, forming arachidonic acid and DHA by genetic variants that adapted to that diet. We have then the derived haplotype. Persons with the derived haplotype are what is called fast metabolizers. They make a lot more arachidonic acid from linoleic and a lot more DHA. But in Africans, it was fixed, and practically everybody in Africa is a fast metabolizer. And then with the transfer of populations, African Americans and others from Africa are fast metabolizers, and they form high amounts of arachidonic acid, which leads them into a pro-inflammatory state, because the frequency of the fast metabolizer genes is much higher, practically 90 or 80% in Africans and African Americans, whereas in Europeans, the derived, the fast metabolizers, as you can see in blue, is about 54%, and the ancestors make up the rest. But Native Americans are very different. Native Americans, they have the ancestral haplotype, and they're slow metabolizers. So unless they eat fish, they are deficient in DHA, in omega-3 fatty acids. And they have the American diet, which is very high in arachidonic acid, omega-6. So we really ought to think about making sure that Native Americans eat enough fish or take fish oils. Now, these high amounts of arachidonic acid and the prevalence of the derived haplotype, they lead to an extremely inflammatory state. And let me now move on into the metabolism of the specialized lipid mediators, which have been shown through lipidomics in very excellent studies the last 15 years, that are potent anti-inflammatory and resolution substances, and they are produced by EPA from omega-3s, DPA and DHA, again from omega-3s. So EPA produces resolvents of the E series, DHA resolvents of the D series, as well as protectants and marasins, and DPA produces resolvents of the D5 series. So these lipid mediators interact with receptors, and they have the following beneficial characteristics. They decrease the cardinal signs of inflammation, which are the skin becoming red, and being hot and swollen and painful. So they decrease the edema and chemotaxis. They decrease the excessive leukocyte infiltration, shown by the red arrow. They decrease pro-inflammatory proteins, and they decrease the cytokine storm, which is prominent in patients with COVID-19. At the same time, they have beneficial effects because they increase the signs of resolution by increasing the clearance of polymorphonuclear cells from the epithelial surface. They increase phagocytosis of the apoptotic PMN cells. There is a removal of inflammatory debris, clearance of bacteria, and there's wound repair and tissue regeneration. In other words, a return to homeostasis. Another way of showing it is showing in red the acute inflammation. And when there is acute inflammation, the very first movement on the part of eicosanoids is the movement and the formation of leukotrienes and prostaglandins from arachidonic acid, which, however, switches very quickly in the presence of lipid mediators to the biosynthesis of lipoxins, which are less inflammatory. That's the only substance from arachidonic acid that's less inflammatory. That leads to resolution because of the biosynthesis of resolvins and protectants from the omega-3 fatty acids, specialized pro-resolvin mediators. They recruit macrophages and they begin to synthesize marasins. And these are pro-resolution effects are achieved, and there is complete resolution and return to homeostasis. So the characteristic bioactions of the specialized pro-resolving mediators coming from omega-3s are pro-resolving agonists, act in pico to nanogram ranges via pro-resolving G protein coupled receptors. Their stereoelective activation of individual receptors, they're anti-inflammatory, and they're immunoresolvents, so they result in homeostasis. And their immunoresolving properties of the specialized pro-resolving mediators is to reduce neutrophil infiltration, enhance phagocytosis, stimulate macrophage efferocytosis, promote tissue regeneration, and control pain. And all of these are formed by the omega-3 fatty acids, alpha-linolenic acid, EPA, DPA, and DHA that are in fish and fish oils. So when COVID-19 pandemic started, it was noted that the people who were more severely affected with COVID-19, there were people who were elderly, over the age of 65, 70. They were mostly African Americans and Hispanics. And there were also people who were obese. You know, obesity is a slow and chronic low inflammatory state, and diabetes, hypertension, and cardiovascular disease. And here is the SARS-CoV-2. And when they did a series of studies to evaluate the various lipid mediators, and in SARS-CoV-2 activated human macrophages, the addition of resolvin D1 and resolvin D2 each dramatically reduce the cytokine storm, diminishing the levels of key inflammatory cytokines, including tumor necrosis factor alpha, interleukin 8, and macrophage inflammatory protein 1. Specialized pro-resolvent mediators reduce eicosanoids that amplify and are mediators of inflammation. These are the ones that are produced from arachidonic acid and are each present in the peripheral blood of COVID-19 patients, along with resolvents that dynamically change with disease severity. So other studies have been done with bronchoalveolar lavages from severe COVID-19 patients. They show high amounts of arachidonic acid produced pro-inflammatory and pro-thrombotic eicosanoids, such as thromboxane metabolites and prostaglandin 2, as well as the potent leukocyte chemoattractant, which is leukotriene B4, compared to healthy subjects, which suggests that their origins in lung tissues of these COVID-19 patients, marking the dynamic procoagulant and inflammatory environment within the lungs of patients with severe COVID-19 disease. It's the high amount of metabolites of arachidonic acid. These lavages also show the presence of lipoxin A4, which is the only metabolite of arachidonic acid that is anti-inflammatory, and many of the resolvents that come in from omega-3, so the D series, notably resolvent D1, resolvent D2, resolvent D4, resolvent D5, and their precursor intermediates. The presence of pro-inflammatory eicosanoids, and specifically resolvents and other SPMs in lung lavages at biologically active concentrations from severe COVID-19 patients suggest that these mediators may have functional roles in the lipid mediator storm of SARS-CoV-2 infections. So in a recent randomized double-blind placebo-controlled study with healthy volunteers, oral administration of marine oil enriched with the precursors of resolvents, 17-HDHA and 18-HIP and 14-HDHA, showed a time and dose-dependent increase in peripheral blood SPMs, likely from endogenous enzymatic conversion of these precursors. In other words, taking fish oils and some of the precursors of the lipid mediators, you have an endogenous increase of the lipid mediators, which can be very supportive. This increase in SPMs correlates with rapid reprogramming of blood cells that include rapid changes in the leukocyte transcriptome and increased bacterial phagocytosis. These changes occur within hours, increasing SPMs that in turn enhance both neutrophil and monocyte ability to phagocytose E. coli, as well as regulate leukocyte transcripts of interest in infections. Hence, the nutritional availability of dietary omega-3 PUFA and marine oils enriched with the SPM intermediate precursors, along with increasing local biosynthesis of SPMs to functional concentrations, may be an approach of value during COVID-19, as well as in prevention and shortening the recovery phase from infections. So the combination of fast metabolism and high omega-6 intake increases their inflammatory status and possibly susceptibility of SARS-CoV-2. In vitro and human studies indicate that the specialized pro-resolvent mediators produced from the omega-3 EPA and DHA influence the resolution of inflammation, allowing the tissues to return to function and homeostasis. The SPMs each counter-regulate cytokine storms, as well as pro-inflammatory lipid mediators via nuclear factor kappa B and inflammasome downregulation, and reduce the pro-inflammatory eicosanoids produced from arachidonic acid. So what are some of the factors that may increase the risk to COVID-19? Increased susceptibility to COVID-19 due to genetic variants in omega-6, omega-3 fatty acid biosynthesis at FADS1 and FADS2 occur at a much higher frequency in African Americans, leading to rapid metabolism of linoleic to arachidonic acid, and then going down to prothrombotic and pro-inflammatory eicosanoids. The same thing occurs in individuals of Hispanic background. About half of the Hispanic population in the US are rapid metabolizers, therefore are in a pro-inflammatory state and are more susceptible to COVID-19. Alaskan natives, they're slow metabolizers, so they don't make that much of either arachidonic acid or DHA. So because they eat a lot of fish, they can balance the high arachidonic acid from the American diet. Native Americans are slow metabolizers, so they're very low in both arachidonic acid and DHA. And I think they definitely are going to need to be told to eat more fish or take supplements. And populations in other parts of the world carry all other kinds of genetic variants, such as intels, insertion deletions in the Chinese people and East Asians that are also lead to faster metabolizers. And the biggest problem, of course, besides the genetic variants, are the high dietary intake of omega-6 fatty acids and the high amount of arachidonic acid in the diet. Poor diet is one of the leading contributing factors for death in the US and worldwide. Unhealthy diets characterized by overconsumption of ultra-processed foods with an omega-6 to omega-3 ratio of 11:1 instead of a balanced ratio have been shown to be associated with increased weight gain, and processed foods and sugary drinks increase the risk of obesity, type 2 diabetes, high blood pressure, and heart disease, all of which, of course, increase the risk to COVID-19. Other factors is age, particularly old age, and as I mentioned before, overweight or obesity, diabetes, cardiovascular disease, hypertension. Pro-inflammatory diets, due to high omega-6 fatty acids, they lead to a hyperactive immune system characterized by increase in cytokines, interleukin-6, which is pro-inflammatory, tumor necrosis factor, which is pro-inflammatory, interleukin-1, which is pro-inflammatory, and C-reactive protein increases that is associated with increased risk in cardiovascular disease. So this type of environment is exactly what SARS-CoV-2, the virus that causes COVID-19, thrives in. So this is a map of the world that shows that there is a world deficiency of DHA with a percent less than 4% in the US and United States and Canada and Brazil and India and the Arab Peninsula, Turkey. The amount that is the amount of DHA that is consistent with health has to be 8%, and you can see that's in green, and it's mostly in Greenland and Iceland and some of the coastal countries. But the arachidonic acid and vegetable oils have taken over, and there is really a worldwide deficiency. So what can we conclude? As we understand the role of genetic variation and through the use of specific nutrients, precision nutrition approaches would predict the physiological and pathological consequences of the interaction of individual genetic differences and diet to prevent and manage adverse outcomes. The emergence of the field of precision nutrition that factors in individual and population-based genetic variability in the context of human diets will provide more specific and individualized dietary and supplement interventions that may prevent and decrease the incidence and prevalence of many of the pro-inflammatory effects of the modern Western diets. Research in the field of precision nutrition will help increase understanding of the therapeutic relationships among food, medications, and a wide range of health conditions and diseases. Research within strategic goal number 4 of the NIH strategic plan, how to improve food as medicine, could have substantial clinical value toward development of standardized and core measures to evaluate clinical nutrition support practices and to determine their effects on health outcomes. Appreciating the contribution of inter-individual genetic differences will be essential for making accurate and effective treatment decisions. It is evident that populations differ in their genetic variants and their frequencies and their interactions with the food they eat. Gene-nutrient interactions is a very important area of study that provides specific dietary advice for individuals and subgroups within a population in the form of precision nutrition. Nutritional science needs to focus on precision nutrition, genetic variants in the population, and a food supply composed of nutrients that have been part of our diet throughout evolution, which is the diet that our genes are programmed to respond. That's not the current Western diet. Thank you.